Other than baby teeth, our organs must last us a lifetime. But any time you look at a plant, you are seeing only a tiny fraction of the plant. You are not seeing all the parts that have already existed and fallen off, nor all the parts that the plant will have in the future. If all those parts could be gathered up and preserved, the total plant would be vastly greater than it ever is at any particular time. Figure 5.22. The cuticle on this ivy has been stained dark pink. The lighter layer is wall material impregnated with cutin. Epidermal protoplasts have stained dark blue. Interior cells are photosynthetic parenchyma. 120. This epidermis was taken from a desert plant, agave. Its cuticle is much thicker than that of ivy and the outer walls of the epidermal cells are extremely thick. Both the wall and cuticle strengthen the leaf surface against insects and prevent water loss. The outer tangential walls are encrusted with cutin, a fatty substance that makes the wall impermeable to water. In species that occur in mild, moist habitats, such incrustation may be sufficient for water retention, but in most plants, cutin builds up as a more or less pure layer called the cuticle. Under more severe conditions, even a cuticle is not sufficient, and a layer of wax may be present outside the cuticle, figure 5.23 and table 5. Cutin and wax resist digestive enzymes and provide defense against pathogens like fungi and bacteria. In some species, the cuticle is so smooth that fungal spores cannot even stick to it and are washed off by rain or shaken off by wind. Waxes are indigestible and non-nutritious, therefore, a thick layer of wax makes it difficult and unrewarding for an insect to chew into a stem. Figure 5.23 on this epidermis, wax is present as flat plates, but in other species it can occur as threads, beads, flakes, or liquid. Unfortunately, cutin and wax also inhibit the entry of carbon dioxide needed for photosynthesis. A totally impermeable epidermis would lead to a plant's starvation. However, the epidermis contains pairs of cells with a hole between them. Guard cells and a stomatal pore together constitute a stoma. Stomatal pores can be opened during the daytime, permitting carbon dioxide to enter the plant. The opening of the stomatal pore is made possible by the structure of the guard cells. Guard cell walls have a special radial arrangement of cellulose microfibrils, which causes some parts to be weaker and more extensible than others. Guard cells swell by absorbing water. The walls next to the stomatal pore are rigid and do not extend, but the back walls are weak and stretch, causing the cells to form an arc and push apart, opening the pore and allowing entry of carbon dioxide and exit of oxygen. This unavoidably allows water vapor to escape and also increases the risk of microbes entering the plant. After sunset, when photosynthesis is impossible, it is not advantageous to keep stomata open. Guard cells in most species shrink and straighten, closing the pore and preventing water loss. Table 5.5 Special chemicals in walls. Figure 5.24 Light microscope view of the epidermis of Zebrina. The stomatal pore, guard cells, and adjacent cells are visible, as are ordinary epidermal cells. Scanning electron micrograph of the epidermis of a cactus showing numerous stomata. Notice how close together stomata are. No parenchyma cell inside the cactus is far from a point where carbon dioxide can enter. The stomatal pore is surrounded by guard cells and then other epidermal cells. Notice the small particle of debris at one end of the pore. Clogging by debris is a problem in dusty areas. In most plants, some epidermal cells elongate outward and become trichomes, also called hairs. Trichomes make it difficult for an animal to land on, walk on, or chew into a leaf. They shade underlying tissues by blocking some incoming sunlight, which may be too intense in summer or in high alpine environments. Trichomes can also create a layer of immobile air next to a leaf surface, which allows water molecules that diffuse out of a stoma to bounce back in rather than be swept away by air currents. Figure 5.25 When guard cells absorb water and swell, they become curved and the inner walls are pulled apart by the expansion of the back walls. If guard cells lose water and shrink, they straighten, closing the stomatal pore. Trichomes exist in hundreds of sizes and shapes. Many are unicellular, being just long, narrow epidermal cells. Multicellular ones may be a single row of cells or several cells wide, and many branch in elaborate patterns. Most trichomes die shortly after maturity, and their cell walls provide protection, but others remain alive and act as small secretory glands. Some secrete excess salt, others produce anti-herbivore compounds, and those in carnivorous plants secrete digestive enzymes onto trapped insects. The poisonous, irritating compounds of stinging nettle are held in trichomes. Cortex. Interior to the epidermis is the cortex. In many plants, it is quite simple and homogeneous, composed of photosynthetic parenchyma and sometimes parenchyma. In other species, it can be very complex, containing many specialized cells that secrete latex, mucilage, or pitch. Some cortex cells contain large crystals of calcium oxalate or deposits of silica. Cortex cells of most plants fit together compactly, but in fleshy stems such as tubers, corms, and succulents, cortex parenchyma is erenchyma, an open tissue with large intercellular air spaces. A few angiosperms, such as water lilies and the plants grown in aquaria, have become aquatic, living submerged in lakes or oceans. These plants have large cortical air chambers that provide buoyancy. The stems have such a strong tendency to float that no sclerenchyma is necessary for support. Figure 526 trichomes. Flat, scale-shaped trichome. Branched trichomes. Glandular trichomes, these are two large single cells. Glandular trichome with a stalk cell elevating the secretory head cell. Glandular trichome, capable of injecting poison. Simple, unbranched, non-glandular trichomes. Figure 527. The cortex of this stem of buttercup is the narrow band of cells between the epidermis and the vascular bundles. The cortex of this corn stem is even narrower, in some places only 2 or 3 cells wide. Vascular tissues. For very small organisms whose bodies are either unicellular or just thin sheets of cells, diffusion is adequate for the distribution of sugars, minerals, oxygen, and carbon dioxide throughout the body. But if any cells of the organism are separated from environmental nutrients and oxygen by just 4 or 5 cell layers, diffusion is too slow, and a vascular system is necessary. 2 types of vascular tissues occur in plants: xylem, which conducts water and minerals, and phloem, which distributes sugars and minerals. Vascular systems of plants are quite different from those of animals. The plant vascular system is not a circulatory system. Water and minerals enter xylem in the roots and are conducted upward to leaves and stems. During its passage, xylem sap travels through dead, hollow cells, not through tubes composed of living cells like our blood vessels. Once in the shoots, most water evaporates from the surfaces of stems, leaves, and flowers and is lost. The minerals and a bit of water are used by surrounding cells. Phloem cells are living. They pick up sugar from areas where it is abundant, usually leaves during summer and tubers or rhizomes in spring, and transport it to areas where sugar is needed, especially growing tips of shoots, roots, young leaves, and flowers. In the later months of summer, phloem carries sugar into developing fruits and into the storage organs of perennial plants. Because sugar must be dissolved to be conducted, water is transported simultaneously in phloem. Xylem. Within xylem are 2 types of conducting cells, tracheids and vessel elements. Both are types of sclerenchyma. The term trachere element refers to either type of cell. As a young cell matures into a trachere element, it first must enter cell cycle arrest and stop dividing. It is initially a small parenchyma cell with only a thin primary wall, but the cell becomes long and narrow and then deposits a secondary wall that reinforces the primary wall. The cell then dies, and its protoplasm degenerates, leaving a hollow tubular wall. Alternatives. Box 5 to 3 simple plants. 3 groups of plants have much simpler bodies than those described in this chapter. Hornworts are a rather rare group of tiny plants whose bodies look like bits of green cellophane, often no larger than 2 to 5 millimeters in diameter and only a few cells thick. They have no epidermis, no stomata, no parenchyma, no sclerenchyma, no cortex, no pith, no xylem, and no phloem. Instead, they are small ribbon or disc-shaped sheets of parenchyma cells, most of which have a single chloroplast. Each cell absorbs water and minerals, carries out its own photosynthesis, and is relatively self-reliant. When the plant reproduces, horn-like structures grow upward, break open, and release spores. During this stage, nutrients are transported from chlorophyll cells to spore-producing cells, but only by means of plasmodesmata, not phloem. Many liverworts are about as simple as hornworts. Plants of Palavicinia, Sphaerocarpos, and Riccia also consist of just small green ribbons or discs of chlorophyllous cells. Bodies of Fossambronia and Pedalophyllum appear leafy and more complex, but in fact, they are just ribbons whose edges are puckered. The bit of complexity present in these bodies is merely that the central portion of the ribbon is several cells thick, whereas the margins are unistratos. As in hornworts, there is no epidermis or vascular tissue. Other liverworts such as Astrella and Marcantia have thicker, more complex bodies. The ventral side is several layers of compact parenchyma with few chloroplasts, and the dorsal side consists of intersecting sheets of cells that form air chambers. The term epidermis is used for surface cells. Dorsal epidermis has stoma-like air pores. Ventral epidermis has trichomes that attach the body to soil or tree bark. The most complex liverworts have leaves and are called leafy liverworts, but their bodies are composed of just parenchyma. As in hornworts, liverworts transfer nutrients through plasmodesmata from chlorophyllous cells to spore-producing tissues when they reproduce. Liverworts have no vascular tissues. Despite their simplicity, all liverworts grow by means of organized apical meristems. Mosses are a bit more complex than either hornworts or liverworts. Mosses always have a stem with leaves, and in those with taller stems or larger leaves, some cells deposit a thicker wall and resemble sclerenchyma fibers. However, these walls do not contain lignin and probably have a separate evolutionary origin, so they are called sterides instead of fibers to avoid confusion. Many mosses conduct water, but along the outside of their bodies, not inside and not with xylem. Their numerous leaves are southwest tiny and close to the stems. They form capillary spaces and act like a wick. A number of mosses do have an internal system of conducting cells in their green, leafy stems. Cells called hydroids conduct water, and others called leptoids conduct carbohydrates and other nutrients. These should never be called xylem or phloem because they differ in significant ways from those two tissues. What are some consequences of having such simple bodies? The simplicity of hornworts, liverworts, and some mosses limits their ability to survive in some habitats. Lacking conducting tissues, they must remain short, thin, or prostrate, and many quickly die in dry air. On the other hand, this same simplicity enables them to survive in many habitats because just a few weeks of cool moist weather is enough to allow their spores to germinate and grow into tiny, mature plants. They do not need to photosynthesize long enough to produce all the carbohydrates that a vascular plant needs for its larger, more complex body. Even deserts have rainy periods and shaded spots that stay moist for weeks. Such spots will almost always have some non-vascular plants thriving there. Figure B5-3a plants of hornworts, the bodies are just thin, green sheets of parenchyma. The columns are reproductive structures. Figure B5-3b plants of this liverwort grow in quiet streams. Each shoot is less than 1 mm thick and consists of parenchyma cells. Each grows from an apical meristem that occasionally divides in two. Figure 5-28 tracheiry elements. Tracheids are long cells with tapered ends. The primary wall is complete over the whole surface, and it is so thin that the inner, spiral secondary wall is visible. The primary wall tore during specimen preparation, revealing just how thin it is and permitting views of the circular bands of secondary wall which are located just interior to the primary wall and which prevent it from collapsing during water conduction. Vessel elements tend to be wider and shorter than tracheids, but the most important feature is the perforation, the large hole at each end. The perforation of one vessel element must be aligned with that of the next if water is to pass through with little friction. The stack of vessel elements is a vessel. The vessel here has been cut down the center, so the front wall and back wall are missing and the vessel appears to be just an open space. 600,270,100. 1,250. Table 5-6 tracheiry elements. The secondary wall is impermeable to water, thus, areas of the permeable primary wall must remain uncovered if water is to enter and leave the cell. In the simplest type of tracheiry element, there is only a small amount of secondary wall, organized as a set of rings, called annular thickenings, on the interior face of the primary wall. This arrangement provides a large surface area for water movement into and out of the cell, but it does not provide much strength. The primary wall must be supported because movement of the water tends to cause it to collapse inward. With helical thickening, the secondary wall exists as 1 to 3 helices interior to the primary wall. Scalariform thickening provides much more strength because the secondary wall underlies most of the inner surface of the primary wall and is fairly extensive. Just as in fibers and scleriids, the area where the secondary wall is absent is called a pit. In tracheiry elements with reticulate thickening, the secondary wall is deposited in the shape of a net, as the name suggests. The most derived and strongest tracheiry elements are those with circular bordered pits. In such tracheiry elements, virtually all of the primary wall is underlain by secondary wall. The pits that allow water movement are weak points in the wall, but the weakness is reduced by a border of extra wall material around the pit, hence the name. Trachyry elements with annular thickenings are weak, but a large percentage of their primary wall is free of secondary wall and available for water movement. Pitted trachyry elements are just the opposite. They are extremely strong, but so much of the surface is underlain by lignifide secondary wall that water enters or leaves the cells slowly. Each is selectively advantageous under certain conditions and disadvantageous under others. If a species grows in perpetually wet soil and water moves easily, its cells are not in danger of collapsing, and thus, trachyry elements with annular or helical secondary walls are adaptive, not only because the large open regions of primary wall allow water to pass through quickly but also because the plant uses little sugar-building secondary walls. If soil dries even slightly, however, water is pulled with more force and exerts inward traction on trachyry element walls. The stronger scalariform, reticulate, or circular bordered pitted walls are necessary to keep the trachyry elements open. Although they cannot conduct water as quickly, water movement is automatically slower because the soil is dry. Under dry conditions, using extra glucose to build stronger secondary walls can be a valuable investment. Figure 529 Trachydes. The growth of a young cytoplasmic cell into a trachyde. After the cell has almost reached mature size, the secondary wall is deposited interior to the primary wall. After the secondary wall is finished, the protoplasm dies and degenerates, leaving only the primary and secondary walls. A trachyde cut open, showing a secondary wall in the form of helical thickenings. A whole cell in which the primary wall is so thin that the annular secondary walls inside are visible. Whole cell with helical secondary wall. Scalariform secondary wall. Reticulate secondary wall. Pitted secondary wall. Light micrograph of a trachyde with a helical secondary wall. Figure 530. As water enters a trachyry element, it passes through primary wall between regions of secondary wall. Water adheres to cellulose molecules, pulling walls inward and upward as it rises in xylem. Primary walls tend to collapse, but secondary walls provide strength to hold the primary wall in place. Under severe water stress, annular and helical secondary walls may not be strong enough to prevent collapse of the primary wall. Trachydes obtain water from other trachydes below them and pass it on to those above. They must occur in groups, lying side by side with some and having their ends overlapping the ends of others. Also, the pits of adjacent trachydes are aligned so that water can pass through. The aligned set of pits is a pit pair, and the set of primary walls and middle lamella between them constitutes a pit membrane, just as in fibers and scleriids. Although very permeable to water, a pit membrane, being a set of primary walls, does offer slight resistance. If each trachyde is 1 mm long, each water molecule passing from root to leaf in even a short plant only 1 meter tall must pass through 1,000 pit membranes. The friction adds up. Figure 531 Side view of a pit that has been bisected. In a scleriid or fiber, the secondary wall would be thicker and the pit narrower and deeper. The diameter of the pit can vary, being broad as in Figure 510D or narrow as in Figure 512. A bordered pit has a rim of extra wall material that prevents the pit from being a weak spot in the wall. With borders, pits can be broad enough to allow rapid flow of water from trachyry element to trachyry element, but not so weak as to be detrimental. Only trachyry elements have bordered pits, fibers and scleriids do not. Figure 532 Bordered pits in face view on front wall of a vessel element. The white slits are the actual openings of the pits, the pinkish area around each white region is the border, and the dark red hexagons are regions where the secondary wall lies against the primary wall. Bordered pits in face view on the front walls of trachydes of pine. This is the same magnification as, notice that trachydes are much narrower than vessel elements, and pits in pine are extremely large. Scalariform bordered pits. Vessel elements provide a way to move water with less friction than trachydes. Vessel elements, like trachydes, are individual cells that produce both primary and secondary walls before they die at maturity. In vessel elements, however, an entire region of both primary and secondary wall is missing. During the final stages of differentiation, a large hole called a perforation is digested through a particular site of the primary wall, often removing the entire end wall. The perforations of adjacent vessel elements must be aligned, and each element must have at least 2 perforations, one on each end. Whereas water must pass through pit membranes each time it enters or leaves a trachyte, it passes through perforations as it goes from one vessel element to the next. Because perforations are wide and completely lack primary walls, they cause little friction. Figure 533. This vessel element was isolated from tissue by dissolving the middle lamella. The secondary wall is clearly visible on the end because the primary wall was digested away before the cell died. These open holes constitute a perforation. The thin, almost transparent covering on the cell's sides is the primary wall, which was not digested. In the plant, another vessel element would have been stacked above this one, with their perforations aligned and permitting water flow. An entire stack of vessel elements is a vessel. The vessel elements on each end of a vessel have only one perforation. Whereas vessel elements, being individual cells, are only about 50 to 100 millimeters long, vessels can be many meters long, running all the way from a root tip to a shoot tip, although some are only a few centimeters long. Vessels must absorb water from parenchyma cells, trachytes, or other vessels, and they must pass it on. Their side walls have pits for this lateral transfer, and all of the types of wall thickening mentioned above for trachytes also occur in vessel elements. The only constant difference is that vessel elements have perforations, complete holes, in their walls, whereas trachytes only have pits. Perforations greatly reduce friction, and water moves much more easily through a set of vessels than through a set of trachytes. Trachytes and vessel elements have plasmodesmata in their primary walls, but these are too narrow to be important in long-distance water conduction. Typical diameters: plasmodesmata, 0.04 micrometers, pits, 1 to 2 micrometers, and perforations, 20 to 50 micrometers. Figure 534. Water passes between trachytes only through pit pairs. Other than plasmodesmata, which are too tiny to be important in water conduction, no holes are present in primary walls of trachytes. Water can pass between vessel elements through perforations, but it can pass from one vessel to another only through pit pairs. Vessels are long, but not as long as a whole plant, and water must, therefore, pass from one vessel to another as it moves upward from roots to stems to leaves. Green equals primary wall, brown equals secondary wall. Within our human circulatory system, all arteries, capillaries, and veins share the same lumen. A water molecule or a red blood cell could circulate through every part of our blood vessel system and never encounter a barrier. In contrast, water molecules must move across pit membranes every time they enter or leave a trachyte or vessel. Some consequences are that the plant system causes more friction than does our vascular system, and solid particles cannot be transported in plants. However, a hole or leak in our system could potentially let all our blood leak out, whereas a hole in a plant system affects only one trachyte or vessel, not the entire system. Trachytes evolved at least 420 million years ago, and virtually all plants with vascular tissue have trachytes. Vessels evolved more recently and occur almost exclusively in flowering plants, where they perform long-distance water conduction in roots and stems. In flowering plants, trachytes occur mostly in the fine veins of leaves but can also be found in the wood of some species. Conifers such as pines and redwoods have xylem composed exclusively of trachytes. Until recently, we thought that ferns too contained only trachytes, but they can at least partially digest their pit membranes and convert pit pairs into sets of perforations. Table 5.7 Length of vessels in trunks of trees. Phloem. Like xylem, phloem has two types of conducting cells, sieve cells and sieve tube members. The term sieve element refers to either. These are very different from trachyry elements. They are parenchyma cells, have only primary walls, and must remain alive in order to conduct. As immature sieve elements begin to differentiate, their plasmodesmata enlarge to a diameter of more than 1 millimeter and are called sieve pores. Plasmodesmata occur in groups, therefore, sieve pores also occur clustered together in groups called sieve areas. Sieve elements remain alive during differentiation, and the plasma membrane that lined the plasmodesma continues to line the sieve pore. The amount of cytoplasm within the pore also increases, and rapid, bulk movement from cell to cell becomes possible. Sieve pores of adjacent cells must be aligned if phloem sap is to pass through. The two types of sieve elements differ in shape and placement of sieve areas. If the cell is elongate and spindle-shaped and has sieve areas distributed over all its surface, it is a sieve cell. This type evolved first and is found in older fossils and in non-angiosperm vascular plants. But because phloem conducts mostly longitudinally, it is most efficient for sieve areas on the two ends of the cell to be especially large and to have very wide sieve pores, whereas sieve areas on the sides of the cell can be rather small. Cells like this are sieve tube members. They are stacked end to end with their large sieve areas aligned, forming a sieve tube. These end wall sieve areas with large sieve pores are sieve plates. Like vessels, sieve tubes are shorter than the plant, and the small, lateral sieve areas are important for transport of phloem sap from one sieve tube to another. Sieve tube members must have evolved at about the same time as the flower. All angiosperms have sieve tube members, but none of the non-angiosperms has them. Figure 535. This fern tracheiry element had digested portions of primary wall out of several pits, converting them to crude perforations. In angiosperms, the wall digesting mechanism is more refined and removes the primary wall more completely. An unusual feature of sieve elements is that their nuclei degenerate but the cells remain alive. Cytoplasm cannot carry on complex metabolism without a nucleus, and in phloem, the necessary nuclear control is provided by cells intimately associated with conducting cells. Sieve cells are associated with albuminous cells, and sieve tube members are controlled by companion cells. These cells are often smaller than the accompanying conducting cell and have a prominent nucleus and dense cytoplasm filled with ribosomes. Walls between conducting cells and controlling cells have many complex passages that are sieve areas on the conducting cell side and large plasmodesmata on the controlling cell side. Companion cells have another important role, that of loading sugars into and out of the sieve tube members. Figure 536. A sieve cell is shaped like a trachide and has sieve areas with sieve pores over much of its surface. A sieve tube member has flat end walls with large sieve pores. On the side walls are only a few small sieve areas with very narrow sieve pores. The micrograph of sieve cells is a high magnification. They are much narrower than the sieve tubes of. Figure 537. Large sieve areas present on sieve plates of sieve tube members. Phloem sap flows from one tube-like sieve tube member into the next through the wide sieve pores. Table 5.8. Sieve elements. Vascular bundles. Xylem and phloem occur together as vascular bundles, located just interior to the cortex. In basal angiosperms and eudicots, vascular bundles are arranged in one ring surrounding the pith, a region of parenchyma similar to the cortex. In monocots, vascular bundles are distributed as a complex network throughout the inner part of the stem. Between the bundles are parenchyma cells. Monocot bundles are frequently described as scattered in the stem, suggesting that they occur at random, but they actually have precise, specific patterns that are too complex to be recognized easily. All vascular bundles are collateral, that is, each contains both xylem and phloem strands running parallel to each other. The xylem of a vascular bundle is primary xylem because it is part of the primary plant body. In addition to conductive trachehides and vessel elements, there is usually a large proportion of xylem parenchyma and even mechanical sclerenchyma in the form of xylem fibers. The vascular bundle phloem is primary phloem, and mixed with sieve elements and companion cells or albuminous cells may be storage parenchyma and mechanical sclerenchyma, usually as phloem fibers, although phloem scleriads also occur. The relative amounts of each cell type within a bundle vary. Storage stems have large amounts of parenchyma in the vascular tissues, vines and tendrils have extra fibers. Figure 538. In a vascular bundle, sieve tube members and companion cells occur together. Companion cells are small and rich in cytoplasm. Sieve tube members are large and appear empty because most phloem sap is lost while preparing the tissue for microscopy. Electron micrograph showing two companion cells and two sieve tube members. The connection between a companion cell and a sieve tube member is a primary pit field with plasmodesmata on the side of the companion cell, but it is a sieve area with sieve pores on the side of the sieve tube members. Cells of primary xylem are larger than cells of primary phloem, and the tracheiry elements on the inner side of each bundle are much smaller than the outer ones. This is true of the primary xylem in all stems, and the reason for this becomes clear when the shoot growth is examined. Stem growth and differentiation. Stems grow longer by creating new cells at their tips. In regions known as shoot apical meristems, cells divide by mitosis and cytokinesis, producing progenitor cells for the rest of the stem. When each cell divides, the two daughter cells are half the size of the mother cell, but they grow back to the original size. As they expand, they automatically push the meristem upward. The lower cells are left behind as part of the young stem. This region just below the apical meristem is the subapical meristem, and its cells are also dividing and growing, producing cells for the region below. In the subapical meristem, visible differentiation begins. Certain cells stop dividing and start elongating and differentiating into the first tracheids or vessel elements of the vascular bundles. Because they constitute the first xylem to appear, they are called protoxylem. Cells around them continue to grow and expand, but soon those just exterior to them also begin to differentiate. Because these had expanded for a longer time, they develop into trachyry elements that are even larger than the first. This process continues until the last cells mature, and this portion of the stem stops elongating. Because these cells have had the longest time for growth before differentiation, they develop into the largest trachyry elements of all, called metaxylem. Figure 539. In this vascular bundle of sunflower, xylem contains rows of vessels alternating with rows of xylem parenchyma. Phloem contains sieve tube members and companion cells. There is a large sclerenchyma cap of phloem fibers. Vascular bundles of monocots have a more complex arrangement. In this corn stem, many large bundles are located near the cortex, and fewer, smaller bundles in the ground tissue. High magnification of a corn bundle, showing primary xylem and phloem. Size is not the only difference between trachyry elements of protoxylem and metaxylem. Because protoxylem elements differentiate while surrounding cells are still elongating, they must be extensible and so must have either annular or helical secondary walls. Although protoxylem cells are dead at maturity, they stretch because their primary walls are glued by their middle lamelles to living, elongating cells that surround them. If protoxylem had scalariform, reticulate, or pitted secondary walls, the cells could not be stretched. Either they would prevent the surrounding cells from growing, or the middle lamelles would break and the cells would tear away from each other. This is not a problem for metaxylem, however, because its cells differentiate only after all surrounding stem tissues have stopped elongating. Because metaxylem cells do not have to be extensible, any type of secondary wall is feasible. A similar process occurs in the outer part of each vascular bundle. The exterior cells mature as protophloem, and cells closest to the metaxylem become metaphloem. Because the sieve elements do not have secondary walls, the walls of protophloem and metaphloem are identical. However, sieve elements are extremely sensitive to being stretched and die when stressed too much. Consequently, protophloem cells are extremely short-lived, often functioning for only one day. Perhaps because they are so temporary, protophloem cells never become well differentiated, their sieve areas are small and rudimentary, and many do not have companion cells. Sieve elements of metaphloem do not differentiate until later, when all surrounding cells have stopped expanding. Metaphloem cells differentiate fully, having large sieve areas and conspicuous companion cells. You might expect metaphloem cells to be very large, like those in metaxylem, but they are much smaller, even though they have undergone long periods of expansion before differentiation. All cells of the region are expanding, whether they are dead functioning protoxylem, dead non-functioning protophloem, or living undifferentiated metaxylem and metaphloem. Cell size differs because cell division is still occurring in some cells but not in others. In xylem, all cells stop dividing, so each cell becomes larger as the tissue expands, protoxylem cells by being stretched and the soon-to-be metaxylem cells by their own growth. Protophloem cells stop dividing and differentiate, however, other phloem cells continue to divide, and they therefore remain small. Figure 540. This shoot tip has several fully expanded leaves, one set of small leaves, and another set of even smaller, younger leaves. Dissection would reveal even more, younger leaves. Longitudinal section through a shoot tip of coleus showing the apical meristem and leaf primordia of various ages. Two axillary buds are just recognizable as small outgrowths. Higher magnification of the coleus shoot apical meristem. Vascular tissue is differentiating in the center of each leaf primordium. This apical meristem of Elodia is much taller and narrower than that of Coleus, and it has many more leaf primordia. This is the giant shoot apical meristem of golden barrel cactus. The meristem is so large it had to be photographed as a collage of approximately 15 smaller images. This image is printed at only 100 times larger than the actual meristem, which is 1.5 millimeters in diameter. Color was altered to enhance the visibility of cell walls and nuclei. Figure 541. Development of three trachyry elements. All cells are young, small, and cytoplasmic. The first protoxylem element forms while all cells are still small. Notice that cell 1 has started depositing its secondary wall. Cells 2 and 3 continue growing, stretching cell 1, which is dead. Cell 2 matures while cell 3 continues to grow, stretching cells 1 and 2. Cell 1 has been stretched so much that it is on the verge of being torn open. All elongation has stopped, and cell 3 differentiates as a pitted element. The primary wall of cell 1 has been torn and no longer conducts. Cell 2 is probably still capable of carrying water. In a stem, these 3 cells would be surrounded by numerous others. Other tissues also differentiate in the subapical region. In the epidermis, the first stage of trichome outgrowth may be visible in the youngest internodes, those closest to the apical meristem. In lower, older internodes, trichomes are more mature, and guard cells and stomatal pores may be forming. The cuticle is extremely thin at the apical meristem, but it is thicker in the subapical region and may be complete several internodes below the shoot apex. Differentiation of the pith typically involves few obvious changes. Cells enlarge somewhat, intercellular spaces expand but remain rather small, and cell walls continue to be thin and unmodified. All changes are usually completed quickly, while the cells are in or just below the subapical meristem. Similar changes occur in the cortex, except that plastids develop into chloroplasts. Protoxylem and protophloem develop quickly while the cells are still close to the apical meristem, but metaxylem and metaphloem do not begin to differentiate until the nodes and internodes have stopped elongating. Because the terms epidermis, cortex, phloem, xylem, and pith typically refer to mature cells and tissues, it is confusing to use them for the young, developing cells during differentiation. To prevent misunderstandings, a special set of terms has been agreed on. Protoderm refers to epidermal cells that are in the early stages of differentiation. Young cells of xylem and phloem are provascular tissues. The equivalent stages of pith and cortex are ground meristem. Thus, the subapical meristem consists of protoderm, ground meristem, provascular tissue, and more ground meristem. The rate of maturation varies greatly from species to species and also from one type of stem to another on the same plant. In stems with long internodes, such as vines, maturation is slow, and immature tissues can be found far below the shoot apical meristem. In stems with short internodes, such as bulbs and lettuce, differentiation may be completed quickly, and all tissues are mature close to the stem apex. Figure 542 cells of an apical meristem give rise to 3 types of subapical cells, which in turn give rise to particular types of mature primary tissues. Epidermis develops only from protoderm, never from provascular tissue or ground meristem. Other cell lineages in the primary body are also quite simple and linear. Now, at the end of the chapter, we can define 2 concepts precisely. Primary tissues are the tissues produced by apical meristems. Primary growth is the growth and tissue formation that results from apical meristem activity. Secondary tissues are produced by different type of meristems and are discussed elsewhere. Plants do things differently. Box 5 to 4 plants and people grow differently. Imagine that your parents planted a tree seed on the day you were born. That tree is probably still getting taller while your growth in height has come to an end. We achieve our maximum height when we are approximately 18 years old, but trees keep right on growing. The tips of their twigs lengthen several inches every spring and make new leaves and flowers. How tall will the tree become? If we treat them well, give them fertilizer, water, sunlight, and protect them from diseases and storms, they may never stop growing. For us, however, our height is set. We will not get to be 7 or 8 feet tall no matter how many vitamins we take, no matter how nutritious our food. We animals have determinate growth. Plants have indeterminate growth. The maximum amount of growing an animal will do in its lifetime, and therefore, its size and the number of organs it has are determined by the genes inherited from its parents. Malnourishment may prevent us from growing properly, but even if healthy and well-nourished, we cannot exceed our genetic potential. The size of most plants is not determined by their genes because even if a plant received genes for poor growth, it might still become extremely large if it lives an unusually long time. An animal's determinate growth is related to its body's precise, invariant form with a specific number of organs. We have 1 head, 1 torso, 2 arms, 2 legs, and our spine contains 33 vertebrae and 12 pairs of ribs, and these were all present when we were born. No new ones were added later. We have determinate organogenesis. Plant bodies are not so precisely organized. When a plant germinates as a seedling, most of its leaves and roots are not present yet, and it has none of its branches, flowers, or fruits either. All of these will be made over the many years of its life, and there is no set number of organs. There will never be a time when it has all of its leaves because some will have already fallen off. Others will not have been made yet. Plants have indeterminate organogenesis. Plants grow by special groups of cells at the tips of their shoots and roots. As these cells divide, approximately half the new cells mature and differentiate into functional cells of the stem, new leaves, and flowers and so on. However, the other cells simply grow back to their original size then divide again. Once again, about half the new cells mature, and the other half continue as cells capable of dividing. The shoot tip always has cells able to generate new cells. There is no reason for it to ever run out of cells. Animals have no set of cells like this, cells always capable of making new cells and organs for the body's growth. Growth is not 100% different in plants and animals, however. Our skin's growth is indeterminate. The innermost layer of our skin, called the stratum basale, consists of cells that never stop dividing. After each round of division, one daughter cell matures as a skin cell, and the other continues to divide. We replace our skin continuously. After a bad sunburn, some skin peels off, but the stratum basale makes more. This is the way plants grow. Our hair, fingernails, and blood also grow continuously from sets of cells that divide and provide new cells throughout our life. Becoming more muscular or fatter is different. That just involves making existing cells larger, not increasing their numbers. Leaf growth in plants is determinate. While a leaf is small, all its cells divide repeatedly. Then, at some point, all differentiate and mature, and the leaf has no cells capable of making any new leaf cells. It expands to its full size as each cell enlarges its central vacuole. The leaf's maximum size is determined by the plant's genes. The same is true for flowers. If we give our plants good care, they may make more flowers but not larger ones. Now for the exceptions, tapeworms have indeterminate growth. They have a head that attaches to the host's intestine and a neck that is a set of cells that divide repeatedly. When enough new cells are present, some organize themselves into a body segment complete with organs. The dividing neck cells continue to make more cells for another segment. The tapeworm makes an indeterminate number of segments, growing to an indeterminate length. Older segments full of eggs break off and end up in the host's excrement, but the neck makes new replacement segments. Unlike other animals, tapeworms do not have a specific number of parts other than one head and one neck. Its plant-like indeterminate growth is based on its plant-like set of permanently dividing cells and plant-like lack of precise body pattern. Exceptional plants are annual plants. These live for just 1 year and, like animals, have determinate growth. Even though they grow at their tip, for some reason those cells all stop dividing after a few weeks or months, ending the plant's organ formation. The plant then blooms and makes fruits and seeds, then it dies. Biennial plants live for 2 years, making many leaves in the first year, blooming and reproducing in the second and then dying. The life of annuals and biennials is programmed by their genes. No matter how much care and fertilizer we give them, we cannot keep them alive longer or get them to grow indeterminately. At the next level. 1. Extreme modification of shoots. All seed plants have the same basic organization of their shoots, but in some, the various tissues are highly modified. Although cacti appear to be exceptional, they have all the normal parts presented in this chapter. You now know enough to understand papers about their anatomy. Parasitic plants that live entirely within their hosts usually have such highly modified bodies that some organs and tissues are missing or barely recognizable. Species you might find informative are Rafflesia arnoldii, Tristerix aphyllus, Viscum minimum, and Hydnora africana. If you really want to go to the next level, look up the family Podostamaceae. These are flowering plants in which almost all aspects of anatomy are abnormal. 2. Evolution and ecology of xylem. All organisms must obtain, transport, and conserve water. Many studies have analyzed the ways in which alternative types of xylem cells affect a plant's adaptation to its habitat. Also the evolution of xylem, from a few simple tracheids in the first plants to the many diverse types of cells present in angiosperm xylem, has been studied extensively and is well documented. Searching on the key words xylem evolution and xylem ecology is a good way to start. 3. Ant plants. A surprising number of plants have stem modifications, such as hollow chambers with tunnels leading to the shoot surface, that provide shelter for animals such as ants and mites. The structures are called domatia, and the insects that occupy them typically provide some sort of benefit to the plant. For example, ants often patrol the plant and kill any other insects that would attack the plant, and some even clean fungi off the plant. Many also urinate and defecate in special chambers of the domatia, thus providing the plant with nitrogen fertilizer. Search on these keywords: ant plant, myrmecophyte, domatium, hypnophytum, myrmecodia, solenopterus. Summary. 1. Cells must be arranged in the proper patterns and with the proper interrelationships in order to function efficiently. 2. All angiosperms have roots, stems, and leaves, although each can be highly modified in particular environments. 3. Virtually all stems function in producing and supporting leaves and transporting water, minerals, and sugars between leaves, buds, and roots. Other stems are involved in storage, reproduction, dissemination, and surviving stress. 4. All stems consist of nodes and internodes, and they have leaves and axillary buds. Numerous types of stems exist. Most individual plants have 2 or 3 types of shoots. 5. The 3 basic types of plant cells, based on walls, are parenchyma, collenchyma, and sclerenchyma. 6. The vascular tissues are xylem and phloem. Trachyry elements consist of vessel elements and tracheids. Sieve elements consist of sieve cells and sieve tube members. 7. Stems of all angiosperms have epidermis, cortex, and collateral vascular bundles. In basal angiosperms and eudicots, the bundles occur as one ring surrounding pith, whereas in monocots, they have a complex distribution in conjunctive tissue. 8. Protoxylem and protophloem form while an organ is elongating and therefore must be extensible. Metaxylem and metaphloem form after elongation ceases. 9. Both primary xylem and primary phloem are complex tissues with a variety of cell types, not just conducting cells. Important terms. Fibers ground meristem guard cells internodes metaphloem metaxylem monocots nodes parenchyma perforation phloem phyllotaxy pits pit membrane pit pair pith. Primary growth primary tissues protoderm protophloem protoxylem pervascular tissues rhizomes scleriids sclerenchyma sieve areas sieve cells sieve plates sieve pores sieve tube members stoma. Trachyry element tracheids trichomes tubers vascular bundles vessel elements xylem. Review questions. 1. The body of an herb contains just 3 basic parts. What are they? 2. Stems of the first land plants function primarily as transport and support structures. Modern stems have these functions also, plus several more. Describe the additional functions. 3. Over the years, several different names have been used for the flowering plants. Here they are called the division, but they are known informally as. There are about species of flowering plants. 4. Early angiosperms diversified into several groups that are now known as. And. Name 3 examples of plants within each line of evolution. 5. An herb has only a primary plant body. That means it has roots, stems, and leaves, but it never becomes and covered with. 6. In general, animals have growth with a fixed size and number of organs, whereas plants have growth with no set size and any number of organs. Which parts of our body grow continuously like plants? Which parts of a plant have fixed size such that no matter how well we take care of the plants, those organs do not become larger? What is the name of plants that live for just 1 year? For just 2 years? 7. What are the important differences between parenchyma, collenchyma, and sclerenchyma cells? 8. Examine Figure 5.6a and 5-6b. Why do most parenchyma cells shown in Figure 5.6a lack nuclei, whereas most shown in Figure 5.6b do have nuclei? In the geranium for Figure 5.6a, did the real cells lack nuclei, or did something happen to the nuclei as the tissue was sliced to make the microscope slide? 9. What is the special name of photosynthetic parenchyma cells? Which organelle is especially abundant in these? How does wall thinness affect carbon dioxide and light? 10. It was previously stated that parenchyma cells are relatively inexpensive to build. What does that mean? How does this relate to most leaves being soft? 11. Like clay, walls of collenchyma exhibits plasticity. Does that mean it can or cannot be stretched? If a tissue is supported by collenchyma, can it still grow? 12. How does parenchyma support a tissue? Would it work if the tissue did not have parenchyma also? What happens if the tissue is not turgid? 13. Parenchyma is plastic, but sclerenchyma is elastic. What does elastic mean? If you stretch or deform an elastic object, will it keep its new shape or snap back to its original shape? As an organ grows, its shape changes, but after it has achieved its mature size and shape, what kind of things would deform it? Should a mature organ have plastic properties or elastic ones? Example, if a heavy load of snow bends a branch. Down, should the branch stay in its bent shape when the snow melts, or should it go back to the shape it grew to before the snow? 14. What are the two types of mechanical, non-conducting sclerenchyma? Which tends to be flexible and useful in wood? Which tends to be brittle and inflexible, useful in pits and stones that protect seeds? 15. Fibers and sclerids have secondary walls that are so thick and tough that the cell cannot grow. If that is true, how do fibers and sclerids grow to their mature size and shape? Do they have a secondary wall even when they are young? 16. Imagine a leaf of a palm tree. When it is fully grown, it must have sclerenchyma to support its size and weight. Do you think it has any sclerenchyma when it is tiny and just starting to grow? Would you expect that it might have parenchyma when it is medium-sized and still growing? 17. Technically, the is an axis, whereas the is the plus any leaves, flowers, or buds that might be present. 18. The point where a leaf is attached to a stem is called a. Just above this point is an bud. 19. Figure 5.13a shows two types of buds. What are the two types and how do they differ? 20. After a leaf falls off a stem, it leaves a just below the axillary bud. 21. What is phyllotaxy? Corn and irises have 2 rows of leaves. This is known as phyllotaxy. 22. When you look at a head of cabbage or lettuce, what are you seeing? Where is the stem? What about when you look at an onion? What are you seeing and where is the onion stem? 23. Describe each of the following types of specialized shoots. Be certain to account for modifications of the leaves, internodes, and orientation of growth, stolon, rhizome, tuber, bulb, corm, and tendril. Each provides a plant with a selective advantage. What is the adaptive value of each type of specialized shoot? 24. Many scientific words are also ordinary English words. If you read that a plant has a bulb or a tuber, can you always be certain exactly what the botanical structure is? 25. What is the outermost surface of an herbaceous stem? The outer walls of this layer are encrusted with a chemical made up of fatty substance that makes the wall impermeable to water. What is the name of the substance and what is the name of the layer? 26. A stoma allows carbon dioxide to pass through the epidermis. What is the name of the 2 cells that control the opening and closing of a stoma? What is the name of the hole itself that carbon dioxide passes through? 27. What is the technical term for a plant hair? Plant hairs make it difficult for animals to do certain things. Name 3 activities that are more difficult for an animal because of a hairy leaf. 28. What is the name of the region of cells between a stem's epidermis and its set of vascular bundles? This is usually a compact parenchyma tissue, but in some aquatic angiosperms that live submerged in lakes or oceans, this region has what type of special modification? Would you hypothesize that this region is especially thick or thin in desert plants such as cacti or plants that have succulent water storage tissues? 29. All flowering plants have 2 types of vascular tissues, which conducts water and minerals, and which distributes sugars and minerals. Are vascular systems of plants similar to or different from those of animals? Explain the similarity or differences. 30. As a young xylem cell matures into a trachyry element, it first must enter and stop. It is initially a small cell, but after it reaches its full size and shape, it deposits a wall that reinforces the wall, the xylem cell, and its protoplasm, leaving a wall. 31. List the 5 types of secondary wall deposition that can occur in trachyry elements. Which 2 types are most characteristic of protoxylem? What is the selective advantage of vessel elements over trachydes? 32. Consider trachyry elements with annular thickening and those with pitted walls. Which is weaker? Which has a large percentage of its primary wall free of secondary wall and available for water movement? Is one always more selectively advantageous than the other? Under all conditions. Explain. 33. Trachytes obtain water from below them and pass it on to above. As water passes into or out of a trachyte, it passes through the set of primary walls and middle lamella in the, is called a. 34. If each trachyte is 1 mm long, how many pit pairs and pit membranes will a water molecule pass through in a plant 1 m tall? Is this a significant amount of friction? 35. What is a perforation in a vessel element? How does it reduce friction as water passes from one vessel element to another? When a water molecule arrives at the end of a vessel, how does it pass into another vessel? In question 34, trachytes were assumed to be 1 mm long. If we assume that vessel elements are also 1 mm long, how many vessel elements are there in an average vessel of American beech? The plant in question 34 was said to be 1 m tall. Would an average vessel of American beech even fit into a plant this tall? If a plant 1 m tall had vessels 1 m long, how many pit membranes would a water molecule need to pass through as it traveled from root to shoot tip? Is this more or less than the answer for question 34? 36. Which evolved more recently, trachytes or vessel elements? Do non-angiosperms such as conifers and ferns have vessel elements? Do flowering plants have trachytes? Vessel elements. 37. Like xylem, phloem has 2 types of conducting cells, and the term refers to either one. Do these die like trachyry elements or do they need to remain alive in order to conduct? 38. What is the name of the holes that interconnect conducting cells in phloem, and what are groups of these holes called? 39. Sieve tube members differ from sieve cells by having very large sieve pores on their end walls, much larger than the ones on their side walls. What is the name of these end wall sieve areas with big sieve pores? 40. Sieve elements lose their nuclei during development, but they must remain alive. What is the name of the cell associated with sieve cells? The one associated with sieve tube members. 41. During differentiation of a young cell into a sieve tube member, what are some changes that occur in the cell wall and the cytoplasm? How long do most sieve tube members live after they become mature? 42. Describe plasmodesmata, pits, perforations, and sieve pores. Which connect living cells? Which connect non-living cells? Which occur in secondary walls? 43. All vascular bundles are. That is, each contains both xylem and phloem. The xylem of a vascular bundle is xylem because it is part of the primary plant body, and the phloem is phloem. 44. Stems grow longer by creating new cells at their tips, in regions known as. Below this region, in the subapical meristem, the very first primary xylem to appear is called, and cells that differentiate into xylem a little later, after they have grown larger, are called. 45. In the apical and subapical region, there are cells that will later give rise to epidermis and other tissues, but they are not yet mature enough to call them epidermis, and so on. What are the terms for the following cells? A. Epidermal cells that are still meristematic. B. Young cells of xylem and phloem. C. Young cells of pith and cortex. 46. Describe the arrangement of tissue seen in a stem cross-section. Consider monocots separately from the other angiosperms. Is the arrangement different in a stolon than in a rhizome, tuber, or corm? 47. Look at figure 542. The top 3 rows are labeled primary growth because these are found in the primary plant body, the body of an herb. The third row of the figure has 5 boxes with the 5 types of mature tissue found in the primary plant body. What are the 5 tissues? 48. If you live to be 100 years old, how many sets of organs will you have during your lifetime? How many hearts, stomachs, and livers? If a plant lives to be 100 years old, how many sets of leaves will it probably have? If it begins blooming when it is 10 years old, how many sets of flowers? When you are 70 years old, you will still depend on your digestive system to supply you with energy for another 30 years. But how old is that set of organs? In a 70-year-old tree, how old are the cells of the leaves that supply it with energy? Design credits: Hummingbird, Tongo 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Marie C. Fields. Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saruri, Plant icon, Shutterstock Incorporated Vector, Digging Man icon, Shutterstock Incorporated Z-Art. Chapter 6 Leaves. Chapter Opener Image. Foliage leaves carry out photosynthesis, which requires that they be exposed to sunlight. But the vine maple shown here is adapted to live in the shade of larger trees in dense forests. In this image, you can see that most leaves receive only dim light and other leaves receive almost no light at all. In contrast, the leaves of the taller trees, the ones that are shading this maple, are exposed to full, intense sunlight. Leaves of different species have evolved to be adapted to extremely diverse habitats. Outline. Concepts. External structure of foliage leaves. Internal structure of foliage leaves. Epidermis. Mesophyll. Vascular tissues. Petiole. Initiation and development of leaves. Basal angiosperms and eudicots. Monocots. Morphology and anatomy of other leaf types. Succulent leaves. Sclerophyllous, foliage leaves. Leaf-soft conifers. Buds callous. Spines. Tendrils. Leaves, with crantz anatomy. Insect traps. Box 6-1 Plants and People, Leaves, Food, and Death. Box 6-2 Botany and Beyond, Leaf Structure, Layer by Layer. Box 6-3 Alternatives, Photosynthesis without leaves. Learning objectives. After reading this chapter, students will be able to list 3 functions of leaves. Identify the parts of a leaf. Compare a simple leaf to a compound leaf. Explain the functions of the epidermis, mesophyll, vascular tissues, and petiole. Name the steps of leaf development. Compare development of monocot and eudicot leaves. Discuss the following leaf types: succulent, sclerophyllous foliage, conifer, bud scales, spines, tendrils, crantz anatomy, and insect traps. Did you know? Some plants, such as lettuce and cabbage, consist almost entirely of leaves, having only a short stem and a few roots. The thin, flat shapes of most foliage leaves are adaptations that facilitate the absorption of sunlight and carbon dioxide. All foliage leaves have chlorophyll. Some plants have red or purple foliage leaves due to such large amounts of red pigment that the chlorophyll is hidden. Leaves have such dense networks of vascular bundles that every leaf cell is close to cells of xylem and phloem. Concepts. The term leaf usually calls to mind foliage leaves, the large, flat, green structures involved in photosynthesis. However, natural selection has resulted in numerous types of leaves that are selectively advantageous because they provide protection, support, storage, and even nitrogen procurement. Because protecting a bud from freezing or drying is very different from photosynthesizing, leaf structures and metabolisms that are selectively advantageous for a bud scale are different from those that are selectively advantageous for a foliage leaf. This principle is true for all types of leaves. For each function, certain modifications are advantageous, whereas others are not. Studying the various leaf types, their modifications, and the roles they play in the plant's biology leads to an understanding of how structure, metabolism, and function are related. The shoot system, containing both stems and leaves, demonstrates both division of labor and integration of distinct plant organs. Stems and leaves must function together if the plant is to survive and reproduce, but the optimal features for each organ are quite distinct. Leaves should be flat and thin for maximum absorption of light and carbon dioxide, and most of their tissues must be alive and differentiated into chlorophyll-rich chlorenchyma to carry out photosynthesis. Stems elevate leaves and conduct material to and from them, among other activities. Maximum conduction and support with minimum expenditure of construction material require a cylindrical stem. Also, much of the stem, all of its trachyry elements and most fibers, must die to be functional. In woody species, new cells are added to the stem xylem and phloem each year, creating a massive wood and bark and an increasingly stronger and more conductive stem. Yearly accumulation of new cells onto leaves is not feasible. Leaves typically live for only several months and are rather lightly constructed. However, in open sunny habitats, elevating leaves on a tall stem is often unnecessary, and a plant may consist almost exclusively of leaves and roots. In lettuce and cabbage, leaves are short-lived and delicate, but in agave and yucca, they are massive, thick, and perennial. Almost all leaves contain only primary tissues. Secondary production of wood and bark in leaves is extremely rare. The initial discussion in this chapter centers on foliage leaves because they are the most familiar. Then other leaf types are described and analyzed. By considering how these leaves differ functionally from foliage leaves, it is possible to determine the environmental factors important to leaf structure and how natural selection has produced diverse classes of leaves. External structure of foliage leaves. The most obvious function of foliage leaves is photosynthesis, but other functions often taken for granted are just as important. Leaves must not lose excessive amounts of water. They must not allow entry of fungi, bacteria, or epifoliar algae. They must not be so nutritious and delicious to animals that they are a liability to the plant. They must not be such effective sails that the plant is blown over in a mild wind, and they must be cheap enough that the plant spends less carbohydrate building them than it recovers by their photosynthesis. If a foliage leaf fails in any of these aspects, the plant dies and the leaf's photosynthesis has been useless. Many structural and physiological aspects of leaves that make them waterproof, pathogen-resistant, and so on in some way interfere with photosynthesis. Although we might assume that foliage leaves are maximally adapted for photosynthesis, these other factors affect leaf biology and must be considered as well. During photosynthesis, leaves absorb carbon dioxide and convert it to carbohydrate by using light energy. Because sunlight comes from one direction at a time, it is best for a portion of the leaf to be flat and wide, allowing maximum exposure to light and maximum surface area for carbon dioxide absorption. Because chlorophyll absorbs sunlight efficiently, light penetrates only a short distance, and thus, leaves can be quite thin. In a thick layer of chlorenchyma, the lowest cells would be in almost complete darkness, unable to photosynthesize. This flat, light-harvesting portion is the leaf blade. The blade's lower side is its dorsal surface, and larger veins protrude like backbones. The upper side is the ventral surface and is usually rather smooth. Figure 6.1 Several possible leaf shapes, all with equal volumes. The common type with a thin, flat blade. All cells are exposed to light, there is a large surface area that absorbs CO, and none of the veins are long. In a thick leaf, light cannot penetrate to the bottom of the leaf, and those cells cannot photosynthesize. Relative to the volume, little surface area is available for CO absorption, but this helps conserve water. A spherical leaf has maximum internal self-shading, and only a small fraction of the cells receives enough light for photosynthesis. Surface area for gas exchange is minimal. A cylindrical leaf could be thin with a large surface area and no self-shading, as in, however, to have the same volume as, it would be extremely long, and conduction through the veins might be difficult. Most leaves have a petiole that holds the blade out into the light. This prevents shading of leaf blades by those above them. Self-shading would defeat the basic usefulness of the leaf. Petioles have other consequences. Long, thin, flexible petioles allow the blade to flutter in wind, cooling the leaf and bringing fresh air to its surface. If the leaf and air are still, carbon dioxide is absorbed from the vicinity of the leaf faster than diffusion brings more to it, depleting the carbon dioxide and decreasing photosynthesis. Leaf flutter also makes it difficult for insects to land on a leaf and knocks off some that have already alighted. Figure 6.2 The petioles of each leaf of poplar are long enough that the leaves do not shade each other, and all are fully exposed to light. The lower side of each leaf blade is covered with white, dead trichomes, the upper surface is hairless and green. Table 6.1 Movement of carbon dioxide. If leaves are small or very long and narrow, self-shading is not a problem, and there may be no petiole. The leaf is then called a sessile leaf instead of petiolate. For example, Aeonium grows in arid, sunny regions, and its fleshy leaves are packed close together. Because the sunlight is so intense, even with self-shading the leaves receive enough light for adequate photosynthesis. Close packing of these leaves helps trap water molecules and prevent their escape from the plant. In many monocots, such as grasses, irises, lilies, agaves, and yuccas, foliage leaves tend to be very long and tapered, and self-shading occurs only at the base. Most of the blade is well exposed to light. They also typically lack a petiole. Instead, the leaf base wraps around the stem to form a sheathing leaf base. The blade can flex and flutter even without a petiole. Not all monocots have linear, grass-like leaves. Palms, aroids, and bird-of-paradise plants have what appear to be petioles and laminas, but the leaves of these species are believed to have evolved from grass-like leaves, and the petiole may actually be a modified portion of the lamina. Figure 6.3 Aeonium tubuliform and its relatives grow in regions of intense sunlight. self-shading is not a problem, but water conservation is. Close packing minimizes water loss from stomata. Figure 6.4 long, narrow leaves are common in many monocots of intensely sunny areas. Shading is no problem, although leaf bases probably do not photosynthesize at maximum efficiency. So tall these are the leaves of St. Augustine grass, the blades project horizontally and are attached to the stem by sheathing leaf bases. In cattails, the sheathing leaf base can be half a meter long and provide firm but flexible attachment to the stem. A leaf blade may be either simple or compound. A simple leaf has a blade of just one part, whereas a compound leaf has a blade divided into several individual parts. Think of how this might be affected by wind. If the blade is either large or flimsy, it twists and flexes and may tear. Tearing can be prevented by making the leaf small or tough or pre-torn. A compound leaf has many small blades, each attached by a petiole to an extension of the petiole, the rachis. Leaves may be palmately compound, with all leaflets attached at the same point, or pinnately compound, with leaflets attached individually along the rachis. Even a large compound leaf has only small leaflets, all of which can flex individually without tearing. Flowing water is even denser than blowing air. Plants that grow immersed in streams typically have compound leaves with thread-like leaflets. Figure 6.5 Maranta, a common houseplant, is a monocot that has broad leaves. Figure 6.6 Simple leaves have numerous shapes, some so deeply lobed as to be almost compound. Poplar and oak have one dominant midrib from which lateral vascular bundles extend. In castor bean, petiolar bundles immediately diverge into several main veins. Figure 6.7 Leaflets of compound leaves have the same structure and metabolism as simple leaves. In pinnately compound leaves, the petioles of leaflets are attached to the rachis, but in palmately compound leaves they attach to the end of the petiole. In either type, any leaflet can be damaged or can abscise without affecting the remainder of the leaf. Figure 6.8 This is a portion of one doubly compound leaf of mimosa. The central rachis bears numerous leaflets, but each leaflet itself is pinnately compound. Compound leaves have other advantages. When a mild breeze blows over two leaves of equal size, one simple and the other compound, it tends to flow smoothly over the simple leaf but turbulently over the more complex surface of the compound leaf. Turbulence brings in carbon dioxide and removes excess heat. Also, an insect can crawl or a fungus can spread across the entire blade of a simple leaf rather easily, but the edges and petiole of a leaflet act as barriers that either prevent or at least slow movement to the rest of the leaf. In some plants, if pathogens severely damage a leaf, it abscisses, carrying the pathogens with it and helping to keep pests away from healthy leaves. Plants and people. Box 6-1 Leaves, food, and death. Leaves impact our lives every day. Examples that come to mind readily are leafy foods such as artichokes, cabbage, celery, lettuce, onions, and spinach. Also important are numerous herbs and spices: basil, bay leaves, marjoram, oregano, parsley, sage, tarragon, thyme, and the flavorings mint, spearmint, and peppermint. The flavors and pungency of these are due to chemicals located within the leaves themselves or in trichomes of the leaf epidermis. Most of these chemicals probably serve as anti-herbivore defensive compounds, causing animals other than humans to avoid the plants. Many classes of anti-herbivore chemicals have evolved, ranging from only mildly effective ones, such as these flavors, to others that are much more powerful, such as alkaloids, many of which are toxic in small amounts and kill quickly. The alkaloids in poison hemlock and death camas are particularly effective. An alkaloid of the leaves of one plant in particular is of interest to us. It is quite lethal but acts only slowly: nicotine in the leaves of tobacco. If tobacco leaves are eaten, they cause vomiting, diarrhea, and even death caused by respiratory failure. However, most tobacco leaves are smoked, of course. Americans smoked an all-time high of 4,345 cigarettes per capita in 1963. Since then, consumption has declined for white males but recently has increased for other groups. Tobacco leaves contain between 0.6% and 9% nicotine, and an ordinary filter cigarette has 20 to 30 milligrams of the alkaloid, approximately 10% of which is absorbed by the lungs. Nicotine dissolves readily into our mucous membranes and passes quickly into our bloodstream. Because it is transferred across the placenta, women who smoke during pregnancy may give birth to babies addicted to nicotine. Blood-borne nicotine also affects the heart, causing coronary problems. People who smoke a pack or more a day are over 3 times more likely than non-smokers to die of heart disease. After nicotine is taken into the cells of the mouth and lungs of a smoker, it can cause cancer, of the lungs especially, but also of the throat, larynx, and mouth. If detected early enough, nicotine-induced lung cancer can be combated with surgery and chemotherapy, but after the cancer has spread to the lymph system, the prognosis is not good. Lung cancer causes more than 400,000 deaths per year in the United States. Figure B61 Basil. Spearmint. Rosemary. Tobacco. Figure 69 Myriophyllum heterophyllum grows immersed in flowing streams. Its leaves are very delicate and pinnately compound with thread-like leaflets. If the stream is quiet, some stems emerge into the air and produce thicker, tougher leaves, hence the species name, heterophyllum. Is it reasonable to conclude that plants with compound leaves are better adapted than plants with simple ones? Why are there any simple leaves at all? Most very large leaves of plants are compound, apparently the only feasible architecture. The large leaves of palms and bananas are formed with a large, single lamina, but they have numerous special lines of weakness and are quickly torn to a compound condition by wind. The largest simple leaves, those of the Victoria water lily, float on water and thus do not need large amounts of sclerenchyma for support, and adhesion to the water's surface prevents wind damage. Among medium and small leaves, simple ones are common, perhaps because a greater percentage of a simple leaf is composed of photosynthetic cells, whereas a compound leaf has a great deal of non-photosynthetic rachis, petioles, and edges. Although most pinnately compound leaves are easily recognizable as leaves, some can be mistaken for a stem with simple leaves. Close examination reveals that leaflets never bear buds in the axils of their petioles, and the tip of the rachis never has a terminal bud. Leaflets are always arranged in 2 rows, never in a spiral, whorled, or decussate phyllotaxy. Some of the tremendous range of shapes of leaves and leaflets is shown in Figure 610. There are hypotheses but no total agreement on how different leaf shapes may be adaptive. Many biologists believe that all function so well that none is clearly superior or strongly disadvantageous selectively. A large number of species, however, have several types of leaves. In the simplest cases, the first few leaves of a seedling are distinctly different from all leaves produced later. If they were merely smaller, it could be argued that the seedling does not have enough stored energy resources to construct the large leaves that a mature plant can afford. But very often leaves of juvenile plants differ from those of adults not only in size but also shape, texture, and even simple versus compound structure. In species such as ivy or citrus, transition from juvenile foliage to adult leaves does not take place until the plant is old enough to flower, perhaps more than 10 years old. It seems reasonable to hypothesize that juvenile leaves of seedlings are adaptive in the microhabitat close to the soil surface where a seedling is located, whereas adult leaves are adapted to the more aerial microhabitat inhabited by an older, larger plant. Figure 610 4 common leaf shapes. Numerous other types also exist. Figure 611 beans have 2 types of leaves. The very first leaf formed by the seedling is simple, but all later leaves are compound. This Azara lanceolata shoot appears to have 2 types of leaves. The larger leaves are true leaves, whereas the smaller ones are actually enlarged stipules. Other species produce 2 types of leaves simultaneously, often one type on long shoots and the other on short spur shoots. In cacti, the spines are short shoot leaves, whereas the long shoot leaves are green, fleshy, and in most species almost microscopic. In a small percentage of species, leaf shape and size are irregularly variable even on one stem, being influenced by environmental conditions that occur at the time of leaf initiation and expansion. Amounts of sunlight and minerals are perhaps the most important factors. Despite numerous possibilities for variation within a species, leaf shape is a valuable tool for plant identification. Overall shape, including leaf base and apex, is important, as is the margin. The margin may be entire, toothed, lobed, or otherwise modified. Within a leaf are veins or bundles of vascular tissue. These distribute water from the stem into the leaf and simultaneously collect sugars produced by photosynthesis and carry them to the stem for use or storage elsewhere. In basal angiosperms and eudicots, they occur in a netted pattern called reticulate venation. In monocots with long, strap-shaped leaves, the larger veins run side by side with few obvious interconnections. This is parallel venation. Figure 612 Several common types of leaf margins. In nature, hundreds of types exist. many being intermediate between two or among several other types. Figure 6.13. A fig leaf held up to the light reveals 5 prominent main veins, numerous lateral veins, and the reticulate venation of all smaller veins in this eudicot leaf. No part of the leaf is without veins. Figure 6.14. These leaves were treated to make their cells transparent, then xylem was stained to make leaf veins visible. In passionflower leaves, veins are narrow and highly branched. At this magnification, the height of the photograph represents 1 mm, and veins occur close together. Leaf veins typically occur closer together than do capillaries in our bones, tendons, and cartilage. Higher magnification of privet leaf. At any point, minor veins are only 2 or 3 trachyry elements wide. These minor veins of crown of thorns are broad and have many wide tracheids at the ends. Acalypha has numerous crystals within the leaf and trichomes on the leaf surface. Figure 6.15. Cattails have long, strap-like leaves common in monocots. Vascular bundles run parallel to each other from leaf base to tip. Figure 6.16. Leaf clearing of corn, a monocot, with several sizes of longitudinal, parallel veins. If a longitudinal vein is broken by insect damage or any other problem, conduction can detour around the site by means of the fine transverse veins. Higher magnification showing the parenchyma cells that surround the leaf bundles in corn. At the leaf base, usually in the petiole, is an abscission zone oriented perpendicular to the petiole. Its cells are involved in cutting off the leaf when its useful life is over. In autumn, as deciduous leaves begin to die, abscission zone cells release enzymes that weaken their walls. As the leaf twists in the wind, cell walls break, and the leaf falls off. Adjacent undamaged cells swell and become corky, forming protective scar tissue, the leaf scar, across the wound. Without an abscission zone, dead leaves might tear off irregularly, leaving an open wound vulnerable to pathogens. In many monocots, ferns, and cycads, an abscission zone is not formed. Instead, a protective corky layer protects living stem tissues, but the dead leaf persists on the plant until it gradually decomposes. Internal structure of foliage leaves. Epidermis. Flat, thin foliage leaves have a large surface area through which water can be lost. Water loss through the epidermis is called transpiration and is a serious problem if the soil is so dry that roots cannot replace lost water. The epidermis must be reasonably waterproof but simultaneously translucent, and it must allow entry of carbon dioxide. Leaf and stem epidermises are basically similar, consisting of a large percentage of flat, tabular, ordinary epidermal cells. Guard cells and trichomes may be abundant. The dorsiventral nature of leaves, however, causes their upper and lower epidermis to exist in significantly different microclimates. On a sunny day, a leaf is usually warmer than surrounding air, thus, it heats the air and convection currents rise from it. If stomata in the upper epidermis are open and losing water, the water molecules are swept away by this convection. However, air tends to be trapped on the underside of a leaf, so water loss from stomata there is not so great. Water molecules are trapped in quiet air and may diffuse back into the stomata. In most leaves, the number of stomata per square centimeter is much greater in the lower than in the upper epidermis. In many species, no stomata at all are found in the upper epidermis. Figure 6.17. Longitudinal section through a leaf axil, showing the abscission zone in the petiole and the axillary bud just above. Magnification of the abscission zone while the leaf is still healthy. The xylem and phloem are not interrupted until the leaf actually falls away. Figure 6.18. As leaves of Joshua tree age, they flex downward and do not abscise. This creates a sheath of tough, fibrous dead leaves with many sharp spines that prevent rats, mice and other small animals from climbing the plant and eating young leaves and buds. Table 6.2. Frequency of stomata in leaf upper and lower epidermis. Such unilateral distribution of stomata has other beneficial consequences. Airborne spores of fungi and bacteria are continually landing on leaves. Rye leaves, for example, typically have 10,000 fungal spores per square centimeter, but most are on the upper surface where stomata are rare, thus, few fungi can penetrate the leaf. Leaf stomata are frequently sunken into epidermal cavities that create a small region of non-moving air. In oleander, the lower leaf epidermis contains numerous crypts, and stomata and trichomes are abundant in the epidermis that lines the crypts. Both the recessed nature of the crypt and the presence of trichomes keep air in the crypt extremely quiet. Many water molecules that diffuse out of open stomatal pores bounce around the crypt and re-enter the stomata rather than being blown away. Leaf epidermises are often remarkably hairy, and trichomes affect leaf biology in numerous ways. What are some of the consequences of having trichomes on leaves? They provide some shade on the upper surface of the leaf, deflecting excessive sunlight, an adaptation common in desert plants. On the lower surface, they prevent rapid air movement and slow water loss from stomata. In any position, trichomes make walking or chewing difficult for insects, and many glandular trichomes secrete powerful stinging compounds that prevent even large animals from eating the leaf. Insects either do not bother with the leaf or must walk so slowly on it that they become more vulnerable to their own predators. Conversely, hairs provide excellent footholds against leaf flutter for insects of the appropriate size. Poisonous glandular trichomes in stinging nettle and other species prevent mammals from eating leaf tissue but also protect leaf-borne insects small enough to walk, feed, and reproduce safely between the stinging trichomes. Figure 6:19 Oleander leaves have stomatal crypts filled with trichomes and stomata. This minimizes air movement near stomata, and water molecules that diffuse out of the leaf may re-enter it by random motion. Crypts occur only in the lower surface. Each stoma of this grass is overarched by 4 protrusions from surrounding cells. This reduces air movement near the stoma. In this bamboo, stomata are protected by protrusions and wax rods. In all 3 examples, the trichomes and waxes prevent water droplets from blocking the stomatal pores. Figure 6:20 Leaves of lamb's ears are so densely covered with hairs that they often appear white. The hairs reduce the amount of heat these leaves absorb from sunlight. Plants of Sempervivum are native to high alpine regions where ultraviolet radiation is intense. The hairs of the leaf epidermis reflect part of the ultraviolet radiation away from the plant. Like stem epidermis, leaf epidermal cells contain a coating of cutin and usually also wax on their outer walls. These retain water and make digestion by fungi difficult. Their smooth, slippery surface prevents spores from sticking or allows them to be washed off by rain. In areas of dew and fog, water droplets condense on trichomes rather than on the epidermis surface. This prevents a film of water from forming across the stomatal pores, which would block movement of carbon dioxide and oxygen until the water evaporated. Mesophyll. The ground tissues interior to the leaf epidermis are collectively called mesophyll. Along the upper surface of most leaves is a layer of cells, the palisade parenchyma, which is the main photosynthetic tissue of most plants. Palisade cells are separated slightly so that each cell has most of its surface exposed to the intercellular spaces. Because carbon dioxide dissolves into cytoplasm slowly, the large surface gives maximum area for dissolution. Tightly packed cells could not absorb enough carbon dioxide for efficient photosynthesis. Palisade parenchyma is often only 1 layer thick, but in regions with intense, penetrating sunlight, it may be 3 or 4 layers thick. In the lower portion of the leaf is the spongy mesophyll, open, loose parenchyma that permits carbon dioxide to diffuse rapidly away from stomata into all parts of the leaf's interior. If stomata were surrounded by closely packed cells, a molecule of carbon dioxide might simply bounce off a cell and escape back out the stomatal pore. Figure 6:21 This leaf of Lorelia has a layer of palisade parenchyma that is 2 cells deep, and the spongy mesophyll is an extensive parenchyma. Figure 6:22 If palisade cells are closely packed with no space between them, only their bottoms are exposed to the carbon dioxide in the spongy mesophyll. With only slight separation of the cells, the volume of the palisade is almost unchanged, but the surface area available is increased enormously. Carbon dioxide can be absorbed much more rapidly. Assume that the palisade parenchyma cells are rectangular columns 20-100 micrometers long. Compute the surface area exposed to CO by 100 cells arranged as inverses. Figure 6:23 And leaves of holly have an extremely thick palisade parenchyma consisting of at least 3 layers of columnar cells. Cells of the lowest palisade layer are relatively widely separated, permitting rapid diffusion of CO from spongy mesophyll to upper palisade 150. Although this arrangement is the most common, some plants have a layer of palisade parenchyma along both leaf surfaces. Spongy mesophyll either occurs in the center or is lacking. The relationship between leaf position and the sun is important. For leaves that are held horizontally, the sun is usually overhead, so having palisade parenchyma near the upper surface permits maximum absorption of light and photosynthesis. For plants that hold their leaves vertically, both sides are equally illuminated, and palisade parenchyma is equally functional on either side. Figure 6:24 Leaves of both carnation and creosote bush have palisade parenchyma along both surfaces. The little spongy mesophyll present is located in the middle of the leaf 75, 150. Vascular tissues. Between the palisade parenchyma and spongy mesophyll are vascular tissues. A eudicot leaf usually has one large midrib, also called a midvein, from which lateral veins emerge that branch into narrow minor veins. Minor veins are most important for releasing water from xylem and loading sugar into phloem, whereas the midrib and lateral veins are involved mostly in conduction. Vein structure changes with size. The midrib and lateral veins always contain both primary xylem on the upper side and primary phloem on the lower side. Because they both conduct and support the leaf blade, they may have many fibers arranged as a sheath, called a bundle sheath, around the vascular tissues. A sheath also makes it difficult for insects to chew into the vascular tissues. Many other types of non-conducting cells such as mucilage, tannin, or starch storage cells may be present in larger veins. Minor veins are the sites of material exchange with the rest of the mesophyll and must have a large surface area in contact with palisade and spongy mesophyll. They do not contain fibers or other non-conducting cells whose presence would interrupt this contact. The endings of the minor veins in some species consist of only xylem and in others only phloem, but they typically contain both. Figure 625 transverse sections through leaf midribs. This midrib of ligustrum is rather simple, containing a single vascular bundle. Xylem is present along the top, phloem along the bottom. A small amount of parenchyma occurs on the top and bottom of the midrib. This midrib of rubber tree has numerous separate bundles embedded in a large mass of mesophyll. This is a midrib prepared as a leaf clearing. The three distinct bundles have no interconnections in this region. Each transports to and from a particular portion of the blade. Veins, especially larger ones, often have a mass of fibers above, below, or both, the bundle sheath extension. Such fibers help give rigidity to the blade and are believed to provide an additional means by which water moves from the bundle out to the mesophyll. Apparently, water moves by capillary action around the fibers rather than through them. Figure 626. This leaf bundle of sugarcane has bundle sheath extensions contacting both the upper and lower epidermis. Water diffuses from the xylem to the epidermis by capillary action, moving around the surfaces of the fibers. The leaves of flax have large bundle sheath extensions consisting of many layers of fibers. Figure 627. And these are minor veins, treated to reveal individual cells. Long, dark cells are trachyry elements, and phloem is out of focus, in line with the xylem. Notice how narrow these veins are. Xylem is only one element wide, so there is maximum surface area for releasing water to the leaf mesophyll. In this species, each minor vein is lined with parenchyma cells that extend outward into the spongy mesophyll. Petiole. Petioles may be tiny but are massive in plants like palms, rhubarb, celery, and water lilies. They are considered to be part of the leaf and are the transition between the stem and the lamina. Consequently, the arrangement of tissues differs at the two ends. The epidermis may be similar to that on the lamina but often contains fewer stomata and trichomes. Petiole mesophyll is rather like cortex, somewhat compact and not especially erenchymatous, and considerable parenchyma is present if the petiole supports a heavy lamina. Vascular tissues are the most variable. 1, 3, 5, or more vascular bundles, called leaf traces, branch from stem vascular bundles and diverge toward the petiole. They may remain distinct or fuse into a single trace at, near, or in the petiole. In some species, they divide into numerous bundles, and 10 or may 20th be found in the petiole. Vascular bundles may either fuse with each other within the petiole or branch further. They can be arranged in a ring, a plate, or a number of other patterns. If the lamina has a strong midrib, most petiole bundles fuse together and form the midvein, but other bundles may enter the lamina as small lateral veins. We do not understand the significance of all the bundle patterns in petioles. Certain patterns may ensure the proper distribution of sugars out of different parts of the lamina, especially of large leaves, into the various bundles of the stem. However, it may be that almost any pattern functions well, and thus, mutations that cause new patterns are not selectively disadvantageous. On the other hand, future studies may show correlations between lamina, petiole, and stem. In many species, the petiole bears two small flaps of tissue at its base called stipules, which serve various functions. They may protect the shoot apical meristem while the leaf is young and small. In other plants, they are large enough to contribute a significant amount of photosynthesis, but usually, when the leaf is mature, the stipules are still small, and they die early. Initiation and development of leaves. Basal angiosperms and eudicots. Leaves are produced only through the activity of a shoot apical meristem. At the base of the meristem, cells just interior to the protoderm grow outward, forming a protrusion known as a leaf primordium that extends upward as a narrow cone, growing so rapidly that it becomes taller than the shoot apical meristem. During this stage, the primordium consists of leaf protoderm and leaf ground meristem, and all cells are meristematic, with dense cytoplasm and small vacuoles. A strand of cells in the center differentiates into provascular tissue and then into primary xylem and phloem, forming a connection with the young bundles in the stem. Botany and beyond. Box 6-2 leaf structure, layer by layer. The internal organization of ordinary leaves is not very complicated, but it can be difficult to visualize from looking only at transverse sections. A series of sections through a leaf of privet is presented here. These are paradermal sections, that is, they are parallel to the epidermis. The first is at the level of the palisade parenchyma, and each successive section is deeper in the leaf. Try to imagine the appearance of the leaf from the perspective of a carbon dioxide molecule. Figure B62 palisade parenchyma. The cells are cylindrical and are not closely packed. Carbon dioxide diffuses easily throughout this layer and is absorbed into the cells through their large surface area. Leaf veins. Just below the palisade parenchyma lies the network of leaf veins. Small lateral veins branch off of larger veins and in turn give off smaller veins. High magnification of a large lateral vein. This section passes through the xylem of the vein, and perforations are visible. Thus, these are vessel elements, not tracheids. These have spiral thickenings. They could differentiate while the leaf was still small, and then as the leaf expanded, they could be stretched and still conduct water. High magnification of minor veins. All mesophyll cells are close to one vein or another. After sugar has been produced by photosynthesis, it does not diffuse very far before it is picked up by a sieve tube member and transported to the stem. Spongy mesophyll. In many leaves, more than half the spongy mesophyll consists of intercellular air space. This allows carbon dioxide molecules to diffuse rapidly into the leaf, away from the stomata. If spongy mesophyll were more dense, carbon dioxide molecules might be trapped near the stomata, then diffuse back out of the leaf before they could be absorbed by a chlorenchyma cell. Lower epidermis. The epidermis is slightly undulate, and as it was cut to make this slide, the knife passed through the center of the cells in some areas and through the outer wall and cuticle in other areas. Stomata are abundant, but the actual stomatal pore is short and narrow. Each is only a small opening through which carbon dioxide can enter. Figure 628. This section of stem has 11 bundles, 3 of which produce leaf traces that enter the petiole. Later, the leaf traces diverge and spread throughout the blade. These traces are drawn without any branching or merging, although both are possible. Cross-section of a petiole of the palm Atalia stained to show its many vascular bundles. Notice how numerous the bundles are compared with the leaves of figure 625. This leaf, like those of most palms, is very large and must have a massive conduction system. In cacti, traces that run to leaves and axillary buds are complex, partly because the cortex is so thick the traces must be long, and partly because leaf and branch traces divide and bifurcate, forming an extensive set of cortical bundles that vascularize the thick, succulent cortex. The central stele of the cactus is the dark material on the right. The dark, thick traces at the top of the micrograph are leaf and bud traces that lead to a flower, and all the others are cortical bundles. Hylocereus undatus. Figure 629. The shoot apical meristem of grape can be seen in 3 dimensions in this scanning electron micrograph. Notice the large leaf primordium. An older leaf primordium was removed. The phyllotaxy is established at the very apex. As the leaf primordium grows upward, it increases in thickness, establishing the bulk of the midrib. A row of cells on either edge of the primordium grows outward, initiating the lamina. As a result of their activity, the young leaf consists of a midrib and 2 small, thin wings. All cells in the wings are meristematic, and their division and expansion enlarge the lamina rapidly. In a compound leaf, 2 rows of loci initiate leaflets, which then grow like simple leaves. During lamina expansion, stomata, trichomes, and vascular bundles differentiate, and the petiole becomes distinct from the midrib. The entire pattern is completed while the leaf is still very small, much less than 1/10 its mature size. In many perennial plants, leaves are initiated in the summer or autumn before they mature. After they reach the developmental state just described, they become dormant, part of a resting terminal or axillary bud. During the next growth period, usually the following spring, the bud opens and the primordial leaves expand rapidly as each cell absorbs water into its vacuole and swells. Little or no mitosis or cell division may occur, only maturation, especially the synthesis of chlorophyll, cutin, and wax. As the leaves expand, the immature, exposed epidermis is very vulnerable, especially to insects. In many species of annual plants, the process is similar, with the embryo in the seed acting like the buds of perennial plants. They initiate leaves before the seed becomes dormant and dry, while it is still inside a developing fruit, and those leaves absorb water and expand rapidly during germination. Figure 630. The uppermost leaf primordium is the oldest, and its sides have begun growing out as the lamina of a simple leaf of grape. In this compound leaf, rather than forming a lamina, 6 leaflet primordia have been established. Monocots. Monocot leaves, like those of eudicots, are initiated by the expansion of some shoot apical meristem cells to form a leaf primordium. Apical meristem cells adjacent to the primordium grow upward along with it, becoming part of the primordium and giving it a hood-like shape. More apical meristem cells become involved until the primordium is a cylinder that completely or almost completely encircles the shoot apical meristem. This tubular portion grows upward as a sheathing leaf base, and the original conical leaf primordium, now located on one side of the top of the tube, gives rise to the lamina. The outer surface of the tube is abaxial epidermis, the inner surface is adaxial epidermis. Meanwhile, the shoot apex enlarges, forms new stem tissue, and initiates the next leaf primordium, which will develop as a tube inside the previous leaf's sheathing base. After the first leaf has grown to its full size, the next leaf continues elongating and emerges through the opening at the top of the previous leaf's tubular sheathing base. Later, another, younger leaf will emerge and so on. Figure 631. Low magnification transverse section through young, developing leaves of the grass Panicum effusum. The central round structure is a section of the shoot apical meristem, and the leaves encircle it. At higher magnification, you can see that all cells in the leaves are still small and cytoplasmic, and the central vacuoles are small. At this stage, all cells are still undergoing mitosis and cytokinesis. Veins are starting to differentiate. At an older stage, leaf veins are more distinct. The first protoxylem trachyry elements are mature in some veins, but most cells that will differentiate into trachyry elements are still enlarging. Figure 632. Buds of buckeye expanding in the spring. Bud scales are being pushed back and will abscise as new foliage leaves swell. The smaller bud on the right contains only leaves, and its apical meristem may be initiating new leaf primordia that will expand a little later. The larger bud on the left has both new leaves as well as flower primordia. In some monocots, the lamina becomes broad and expanded like a eudicot lamina, but grasses, lilies, and many others have linear, strap-shaped leaves that grow continuously, having no predetermined size. Their lamina grows by a meristem located at its base where it attaches to the top of the sheathing leaf base. The lamina's meristematic cells remain active mitotically, producing new cells that extend the leaf. Even if a grazing animal, a range fire, or a lawnmower destroys much of the leaf, the meristem forms more lamina. This type of regeneration is not possible in most leaves. The constant basal expansion in monocot leaves means that protoxylem and protofloem are constantly being stretched and disrupted in the basal meristem. New vessel elements and sieve tube members differentiate rapidly enough that conduction is never interrupted. Just above the basal meristem is a region where tissues differentiate, more primary xylem and phloem are initiated, as are stomata and other features. Higher above the basal meristem, tissues are mature. Differentiation is similar to that for stems but is oriented upside down in monocot leaves. Figure 633. Aphids and other sucking insects typically attack young leaves and stems that have not yet developed any protective sclerenchyma around the vascular bundles. Figure 634. In a monocot, after the center of the leaf primordium has been initiated, neighboring regions of the shoot apical meristem become active and contribute to the primordium. This results in a leaf base that encircles the stem, forming a sheath around it 150, 120. The leaf blade of this dendrobium orchid has developed from a leaf primordium tip that resembled the tip in, and the sheathing base here developed from elongation growth of the small collar of leaf primordium tissue visible in. Alternatives. Box 6-3 photosynthesis without leaves. Photosynthesis may occur in tissues other than foliage leaf mesophyll, for example, stem cortex and bark. Carrying out photosynthesis in these alternative sites has particular consequences. Bark. A small number of trees produce green, chlorophyllous photosynthetic bark. This is rare and occurs almost exclusively in desert-adapted trees that abscise their leaves during dry periods, after which they must rely entirely on their bark for sugar production. The trees, often called palo verde, produce cork cells in their bark like all trees do, but they also produce cells called phytoderm, which contain chloroplasts. Phytoderm is usually rather compact with few intercellular spaces, and very little of the cell walls are free surface, that is, surface that faces intercellular space and that can absorb carbon dioxide. Consequently, carbon dioxide neither diffuses rapidly through phytoderm nor is it absorbed quickly. Furthermore, the layer of phytoderm cells is covered by several layers of cork cells, which are impermeable to gases and thus block uptake of carbon dioxide. The green bark of these trees probably carries out recycling photosynthesis. All living cells of the tree trunk and branches respire and give off carbon dioxide just like we animals do, and the green bark can recapture it and convert it back to carbohydrate. In trees with non-photosynthetic bark, that carbon dioxide would simply leak out of the bark and be lost. Cortex. Young stems of almost all plants have a chlorophyllous cortex. Most stems are too narrow to provide much surface area and thus capture little light. Whereas stomata are virtually always present in foliage leaf epidermis, they may be lacking in stem epidermis. And if so, little carbon dioxide can be absorbed. Even if stomata are present, stem cortex, like phytoderm, is typically more compact than leaf mesophyll and thus does not permit rapid diffusion or uptake of carbon dioxide. Stem succulent plants. Many desert plants store water in enlarged, chlorophyllous stems. Stem succulents may produce large, thin, ordinary foliage leaves during the desert's brief moist period, abscising these leaves when drought begins, or they may produce only very tiny leaves. Either way, stem succulent plants usually depend on their chlorophyllous cortex for much or all their photosynthesis, and their cortex typically has many leaf-like features. Their stem epidermis has a high density of stomata, sometimes having as many stomata per square centimeter as leaf epidermis. The outermost layer or two of cortex cells may be very aerenchymatous, creating a tissue similar to spongy mesophyll, allowing carbon dioxide to diffuse rapidly away from stomata and deeply into the cortex. Chlorophyllous cortex often resembles palisade mesophyll, consisting of long columnar cells. Whereas palisade mesophyll is only rarely more than 1 or 2 cell layers thick, palisade cortex in stem succulents is often 5 to 10 cell layers thick, being as much as 1 to 1.5 millimeters thick. Any photosynthetic tissue must be well vascularized, but cortex typically does not have its own set of vascular bundles. This vascular bottleneck can be overcome by several alternative arrangements. If the cortex is narrow, its chlorenchyma cells are close to the stem's vascular bundles. If the stem has short internodes, then leaf traces are also close to each other and provide some vascularization to the cortex as they run out to the petiole base. The cortex of cacti is exceptional in that it does have its own particular vascular system called cortical bundles. This is a network of collateral vascular bundles that branch and anastomose, forming a three-dimensional reticulate venation. Cactus cortex is well vascularized, and no matter how rapidly it photosynthesizes, phloem cells are close by and can quickly load the sugars and transport them away. Figure B6-3 trunks and branches of trees called palo verde are green because their bark contains cells with chlorophyll. No stomata are present, so no new carbon dioxide can be absorbed from the air. Trunks and branches of saguaro cactus are green because the epidermis is still present and transparent, allowing us to see the green of the cortex cells inside. Stomata are present, and saguaros do bring in carbon dioxide from the air and convert it to carbohydrate. As a saguaro absorbs water after a rain, its parenchyma cells swell so the entire stem swells, the ribs flatten out and the epidermis is not torn. If it does not rain for many months, the cactus gradually loses water, despite its cuticle and wax, and shrinks. The ribs also shrink and become narrower. A ribbed body like this can change volume without changing surface area. Broad succulent stems store water, which causes them a problem not usually faced by foliage leaves: how to allow volume to increase while surface area remains constant. We animals generate new skin anywhere, anytime, which is a good thing. If we become overweight, our volume increases, as does our surface area, and our skin grows rather than tearing apart and revealing our insides. However, plants only generate new epidermis at shoot and root apices and in growing leaves and flowers. Stem succulents solve this problem by having an accordion-like, pleated surface. As water is stored and volume increases, ribs become wider; as water is lost, ribs become narrower. Volume changes dramatically but surface does not. In these alternative photosynthetic systems, water conservation is an important factor, so none of these alternatives is free to evolve to be as effective at photosynthesis as an ordinary foliage leaf is. Most modifications that increase water conservation decrease photosynthesis and vice versa. In moist habitats, water conservation is of little importance, so modifications that increase photosynthesis will increase survival of the plants even though they cause plants to lose water more rapidly. In contrast, modifications that conserve water in dry habitats are more valuable than those that allow maximum photosynthesis. Of course, desert plants are not free to maximize water conservation to the point where they do not photosynthesize at all. Morphology and anatomy of other leaf types. Succulent leaves. Numerous adaptations permit plants to survive in desert habitats, one of the most common being production of succulent leaves. This is characteristic of species in the families Crassulaceae, Portulaceae, and Isoaceae, among others. Succulent leaves are thick and fleshy, a shape that reduces the surface-to-volume ratio and favors water conservation. Some leaves are cylindrical or even spherical, the optimal surface-to-volume shape. The reduction in surface area, which is advantageous for water retention, has the automatic consequence of reducing the capacity for carbon dioxide uptake. Inside the leaf, the mesophyll contains very few air spaces, reducing the internal evaporative surface area and, in turn, water loss through stomata. A lack of air spaces also makes the mesophyll more transparent, just as pure water is more transparent than soap bubbles or foam, allowing light to penetrate farther into the leaf. Photosynthesis occurs more deeply than it would in the foliage leaves described earlier. In some members of the genera Lithops and Frithia, leaves are so translucent they act as optical fibers. The leaves are located almost completely underground, where it is cool and relatively damp. The exposed leaf tips allow sufficient light to enter and be conducted to the subterranean chlorenchyma. Although the plants live in a harsh desert in Madagascar and southern Africa, photosynthesis actually occurs in a rather mild microclimate. Figure 635. Leaves of Senecio rotundifolia are spherical, giving them an optimal surface-to-volume ratio for conserving water. Leaves of the succulent Dinteranthus are hemispherical and attached to the opposite leaf, greatly reducing the exposed leaf surface. Interior tissues are mostly water storage parenchyma. Their low levels of chlorophyll result in low levels of photosynthesis, but a lack of water is a more significant danger for these plants. Each plant consists basically of two leaves and a microscopic stem. The root system is substantial. Dinteranthus and many related species have only two leaves at a time. As two new leaves expand, the existing two collapse and wither away. This agave has survived a wildfire in West Texas because its succulent leaves protected the stem from the heat of the fire. The center of the plant, and especially the shoot apical meristem, is still healthy. Containing so much water, the leaves heat up slowly, and before the innermost leaves became hot, wind had blown the fire farther on and the stem and apical meristem were saved. The leaves even kept the ground at the base of the plant cool, protecting the roots. Figure 636. Lithops, stone plant, is closely related to Dinteranthus. Both are in the family Isoaceae. Lithops also has only two leaves, but they are located almost entirely underground. The flat, translucent tips project above the soil and conduct light to the subterranean chlorenchyma. Sclerophyllous foliage leaves. Foliage leaves must produce more sugars by photosynthesis than are used in their own construction and metabolism, or the plant would lose energy every time it produced a leaf. This limits the amount of sclerenchyma in foliage leaves, and most leaves therefore tend to be soft, flexible, and edible. In some species, leaves have evolved which are perennial, existing on a plant for 2 or more years. With this extended lifetime and prolonged productivity, sclerenchymatous leaves are feasible, and their hardness makes them more resistant to animals, fungi, freezing temperatures, and ultraviolet light. Such plants are sclerophyllous and the leaves are sclerophylls. The sclerenchyma is often present as a layer just below the epidermis and in the bundle sheaths, although the epidermis itself can be composed of thick-walled cells. The cuticle is usually very thick, and waxes are abundant on leaves of many sclerophyllous species. Figure 637. Leaves of barberry are tough and hard. It is difficult for insects to bite into them or lay eggs in them. However, the plant must invest considerable glucose to make the secondary walls of sclerenchyma cells, and the leaves must therefore photosynthesize longer before they reach their break-even point. Leaves of conifers. In almost all species of conifers, leaves are sclerophylls. They have a thick cuticle, and their epidermis and hypodermis cells have thick walls. Most conifer leaves contain unpalatable chemicals. Conifer leaves are always simple, never compound, and have only a few forms. Needles, either short or long, occur in all pines, firs, and spruces. Needles of longleaf pine can be 40 centimeters long, although most other species of pine have needles about 10 centimeters long. Small, flat, scale-like leaves form a shield-like covering on stems of junipers, cypresses, arborvitae, and others. In Agathis, Araucaria, and Podocarpus, all genera of the southern hemisphere, leaves are rather large, broad scales held away from the stem. Lengths up to 12.5 centimeters and widths of 3.5 centimeters have been measured in leaves of Podocarpus wallachianus. Conifer leaves are mostly perennial, remaining on the stem for many years. Consequently, the plants are evergreens. Needles of bristlecone pine live for at least 5 years. Their vascular bundles can produce new phloem each year, but no new xylem. The small-scale leaves of juniper also persist and are photosynthetically active for many years, and leaves of agathis and araucaria remain even on very old trunks. 3 conifers have annual leaves that are shed each autumn. Larches, bald cypress, and dawn redwood are all deciduous. Bud scales. One of the most common modifications of leaves is their evolutionary conversion into bud scales. In perennial plants, dormant shoot apical meristems are protected from low temperatures and the drying action of wind during winter by bud scales, which form a tight layer around the stem tip. Because their role is primarily protection, not photosynthesis, bud scale structure differs from that of foliage leaves. Bud scales are small and rarely compound, so mechanical wind damage is not a risk for bud scales. Their petiole is either short or absent because they must remain close to the stem and be folded over it. To be protective, they must be tougher and waxier than regular leaves. Bud scales frequently produce a thin layer of corky bark, at least on exposed portions, which provides greater protection than the simple epidermis of foliage leaves. Spines. Cacti have 2 types of leaves. The green cactus body has microscopic green leaves, and the clusters of spines are their axillary buds. Cactus spines are modified leaves of axillary buds. The succulent, moist cactus body would be an excellent source of water for animals were it not for the protective spines. As with bud scales, spines have a distinct structure related to their function. The soft, flexible blade of a photosynthetic leaf is useless as a protective device against herbivores, but spines have no blade and are needle-shaped. Mutations that inhibit lamina formation have been selectively advantageous. No mesophyll parenchyma or vascular tissue is present. The mesophyll instead consists of closely packed fibers. After fibers mature, they deposit lignin in their walls, which makes them hard and resistant to decay. The cells then die and dry out, hardening even further. Because cacti carry out photosynthesis in their stem cortex, loss of the leaf lamina was not selectively disadvantageous, but for plants that have no alternative photosynthetic tissues, mutations that cause loss of lamina are extremely disadvantageous. Spines of many other species have a similar structure. Figure 638. Yucca leaves are excellent examples of sclerophylls. They are tough and fibrous, and the cuticle is thick. These features reduce the amount of light reaching the chlorenchyma, but plant growth is probably limited by scarcity of water rather than reduced photosynthesis. These leaves may function for 5 to 10 years or more. Bundles of fibers occur just interior to the epidermis, and the thick mesophyll surrounds many vascular bundles. Stomata occur in long, shallow grooves that run between fiber bundles. Even vascular bundles of these sclerophylls have fiber sheaths. Sclerophylls of Dracaena are very thick and have leaf veins throughout the mesophyll, unlike the single row in ordinary leaves. Leaf veins of Dracaena have extremely thick fiber sheaths. Figure 639. Many conifers, such as this lodgepole pine, have needle-shaped leaves. Incense cedar and many other conifers have scale-shaped leaves. Figure 640 scale-shaped leaves in monkey puzzle tree are very large and persistent, living for many years, even as the branch enlarges and becomes woody. Figure 641 needle leaves of pine are round in transverse section. Needles last for many years, producing more phloem each year. Pine needles are sclerophylls, they have thick-walled epidermal cells with a thick cuticle. They also contain resin canals. Figure 642 bud scale